In this, we'll study about different microbes and all that are, or the human diseases, about all that we will study. So the chapter carries a total of 12 marks. So first question that will be asked in your practical exam is isolation of the DNA. So it carries a total of how many? 5 marks. So you yourself will have to do the experiment in a laboratory. Question paper is divided into 5 sections, that is A, B, C, D and E. Part A, Part B, Part C, Part B and Part E. Hello everyone, a warm welcome to this introductory class on biology for class 12th CBSE. I'm Dr. Divya, biology faculty. So we'll go with the introduction to the syllabus. So in this particular session, we'll talk about the scope of the syllabus. So that is what all are the contents present in the syllabus, and we'll look into what are the textbooks used for reference as well as the lab manual that is needed. And we'll go on for knowing about the course structure. Then we'll talk about the question paper design for the practicals as well as the theory. So talking about the textbooks. So textbooks that are usually preferred for class 12th biology is the NCERT biology textbook. That is the National Council of Educational Research and Training. They have framed textbooks which have been followed since many years. So the same textbook has to be followed and the same contents which are there in the textbook needs to be taught. And the lab manual is also being released by the NCERT. So the practicals will be conducted based on the experiments that are given in this particular lab manual. Next, moving on to the course structure. So the syllabus mainly consists of 5 units. So here 5 units, I've started it from unit 6, 7, 8, 9 and 10. First to 5th units that were there that you have studied in your 11th biology. So here the remaining it is a continuity, wherein 6th unit, it is about reproduction. So reproduction, it carries a total of 16 marks. That is in the question paper that is set for CBSE biology. A total of 16 mark questions, be it assertion reasoning, multiple choice questions, be it competency based questions or direct questions, analysis kind of question, all that will come, which in total weightage for the unit 6, that is reproduction, is 16. Next, moving on to the next unit, that is the 7th unit, it is genetics and evolution. So in genetics and evolution, we'll study about the Mendel's laws of inheritance or the dihybrid cross, the monohybrid cross, and we'll study about Darwinism, his theories, the evolution, how it took place, all that will be studied under this. So that chapter or the whole unit that is there, or what all chapter comes under that, total weightage for that unit is nothing but 20 marks. So next, moving to the 8th unit, it is about biology and human welfare. So in this, we'll study about different microbes and all that are, or the human diseases, about all that we will study. So the chapter carries a total of 12 marks weightage for that. The next unit is about biotechnology and its application. Under this, we'll study about the recombination technologies, the replications of the DNA and the mRNA strands, how it gets formed, or what are the different biotechnologies and its field of applications, all that we will study under unit 9. And this unit carries a total of 12 marks. So for 12 marks from this unit, questions will appear in the exam. And the last unit is ecology and environment. Under this, we'll study about biodiversity and conservation, about the ecosystem, about the ecology and all that. And it carries a total of 10 marks. So totally your theory paper will be for 70 marks, wherein from unit 1, you'll get questions for a total of 16 marks. Next unit, it is 20, 12, next 12 and then 10. So overall, your theory paper you'll be writing for 70 marks. So remaining, that is 30 marks is for your practical. So this is how the course structure. So among these units, the maximum questions will appear from, or the maximum marks will appear from unit 2, that is genetics and evolution. So it carries a total weightage of 20 marks. So moving further, let's look into what is the syllabus that is prescribed by the board, or what is the syllabus that is there in the NCERT textbook. So here talking about unit 6, which is mainly about reproduction. In this reproduction unit, we have to study about the reproduction in the flowering plants as well as in humans. So here we need to learn about the sexual reproduction in flowering plants. So actually chapter 1 is being deleted since the last year. So you have to study about from chapter 2. So sexual reproduction in flowering plants. Then we have to move into chapter 3, wherein we'll study about the human reproduction, the male and the female reproductive system. And chapter 4 is about reproductive health. So what all needs to be done to maintain a proper reproductive health for fertility and all that, what needs to be done. Next, in unit 7, wherein I told you it's about genetic and evolution, under that, chapter 5, it is about principles of inheritance and variation, wherein entirely you will be studying about Mendelian genetics, like what are other laws given by Mendel and how did he come across the dihybrid cross, the monohybrid cross, the test cross, back cross, and all that you will study under this. Next, under chapter 6, it is about molecular basis of inheritance. Under this, you will study about the DNA, its replication process, what are the nitrogenous bases present in the DNA, the structure of those nitrogen or the components of those nitrogenous bases, all that you will study under molecular basis of inheritance. And you will study about some genetic disorders also under this, mainly related to chromosomes is what we study under chapter 6, that is molecular basis of inheritance. Next, chapter 7 is entirely about evolution, wherein we study about the Darwin's theory of evolution, Lamarck's theory of evolution, about the giraffe phase, and about Darwin's voyage is all that we study under, and how the Earth, the present gaseous environment that is there, how did it develop over years is what we study under chapter 7, that is evolution. So that is what we study under evolution. Next, moving to unit 8, it is about biology and human welfare. So in this, we study about the human health and diseases. Chapter 8, human health and diseases, various microbes, though they are useful for the human beings, there are some microbes which cause a lot of problems to the human health through various diseases. Under chapter 10, we learn about microbes in human welfare. I told you, lots of microbes are pathogenic, wherein they cause a lot of diseases in humans. So that we'll study under chapter 8. In chapter 10, it is entirely dedicated to the good microbes. So we have lactobacillus and all that, which are very, very important for the human being. So various microbes are of great industrial importance or commercial importance. So about example, yeast is one of the example. So about all that, we'll study under chapter 10. So moving further, under unit 9, we'll study about biotechnology and its application. So in this, we'll study about various principles and processes that is involved in biotechnology. Say, for example, we have the recombinant DNA technology, about all that we'll study here. And under chapter 12, it is about biotechnology and its application. So biotechnology or genetics has been applied in various fields. What are they? How is it useful? And which fields they are applied for? So all that we will study under chapter 12, that is biotechnology and its applications. Next, moving to the last unit, that is unit 10, it is about ecology and environment. So ecology and environment, chapter 13, it is about organisms and population. So what are the different organisms, how population density is calculated, the birth rate, the death rate or mortality, all that, infant mortality, water, all those terms will study under this particular chapter. Next, chapter 14 is entirely dedicated to ecosystem, wherein we'll study about the different forms of the ecosystem. Chapter 15, it is about biodiversity and the important biodiversity hotspots and how to conserve the biodiversity is what we learn under chapter 15. So this is about the entire syllabus. So almost all the chapters are quite interesting under class 12th biology. So talking about, so this was about the course structure for theory. So theory, we got to know that from which unit, for how many marks the questions will be asked and what is the weightage of each of these unit. And we got to know that we are writing the theory paper for a total of 70 marks. So remaining 30 marks is for the practicals. So practicals, that is the experiments that we conduct in the lab. So you'll have separate laboratory classes done for that. In that, there is a course structure that is there for practicals. So for practicals, you'll have the question paper will be like this. So wherein you'll have one major experiment which you'll have to perform and you'll have to write about it. So it will carry a total of 5 marks, and experiment number 5 will come for the first question in your practical exam. So experiment 5 is nothing but isolate the DNA from available plant material. So DNA, by various techniques, we can isolate the DNA, wherein in the laboratory you add certain chemicals, you grind the material, then you add certain chemicals to it, and you can get spools of DNA from it, which you'll have to do in the laboratory and show the DNA thread or the DNA spool to the examiner. So we will be using different materials here like spinach, green peas, then we have papaya, all that we will use to extract the DNA content, because at the end of the day we can use any material, but these are the ones which will give a good result. So that is why these are preferred. So as I told you, one major experiment, that is the 5th experiment. What is the 5th experiment? So first question that will be asked in your practical exam is isolation of the DNA. So it carries a total of how many? 5 marks. So you yourself will have to do the experiment in a laboratory. Next question is one minor experiment, that is experiment 2 and 3. It carries a total of 4 marks. So experiment 2 and 3 is this one. So this is question 1. So this is question 2. This also is question 2. Now will both be asked for you? No. It will be a chit system. So you'll have to pick it up. So which experiment comes to you, that you'll have to do out of the 2. So which experiments you will get? You'll get study the plant population density by quadrant method. So quadrant method is nothing but a square. Say for example, there is a forest. There are n number of trees there. Can you just stand in one place and count the trees as such? No, not possible. Okay, can you go inside and from one corner to the other corner, can you count the trees? No. Why? Because the trees are not planted in the forest in a straight line. It is haphazard. So there what they do is they make a square with a thread. They tie it from one tree to the other. They make a square boundary. And whatever tree falls in that boundary, that is counted. So bit by bit, they go on counting like that. So that is called as the quadrant method. So that should be practically shown in the form of a chart and all that. Through charts you can do it and show it to the examiner how it is done. Next is study the plant population frequency by quadrant method. Now any species of plant, how many numbers of those species are there in that particular quadrant? That is what you need to calculate. So that is the next experiment. So your minor experiment, your major experiment is isolation of DNA. Your minor experiments are study plant population density by quadrant method and study plant population frequency by quadrant method. So any one you will get, which you'll have to do in the exam. Next question in the practical exam is to prepare the slide. So you have to prepare the slide, which will carry a total of 5 marks. So your preparation of the slide will carry few marks, and the theory part that you write for it will carry few marks. So under this, question number 1 and 4. So 1 and 4 is what is that? For question 3. These two will come for question 3. So prepare a temporary mount to observe pollen germination. So for pollen germination, we can take vinca flower and we can dust the anther onto the slide, and then we have to add somewhere about 10 to 20% of sucrose solution, which will already be prepared in the laboratory, and we have to wait for 15 to 20 minutes depending on the room temperature, depending on the weather, and observe under the microscope for germination. So the pollen tubes will be produced out of the pollen grain. So that is what we need to observe under this. And next is prepare a temporary mount of onion root tip to study mitosis. So already the prepared material will be given to the student, that is the onion root tip. The student has to squash it and search for the stages and focus any one stage and show it to the examiner. So in your 11th, you have studied the different stages of mitosis, right? So we studied prophase, metaphase, anaphase, telophase. So out of these 4, any one stage you need to show it to the examiner. So that is question 3. So this is also a minor experiment, that is slide preparation 1, question number 1 and 4. So it carries a total of 5 marks. Next, 4th question is entirely for spotting. Here you don't have to do anything. The material will be kept in the place. You have to identify what it is. For identification, you will get 1 mark, and writing points on it, you will get somewhere around 6 marks. So therefore, it will carry a total of 7 marks. So for spotting, there are different experiments for spotting. Say flowers adapted to pollination by different agencies, that is by wind, insects, then birds and all that. So here, say for example, they'll keep a photography of a flower wherein the stigma is feathery. So if the stigma is feathery, it is understood that it is wind pollination. So that the feathery stigma will just sway in the air like this, and whenever the pollens are traveling in the air, it will catch that pollen. Okay, so it is understood it is by wind pollinated. Or insects, say for example, the stigmas will be very long, the anthers will be very long, they will be exposed out of the flower, so that the insects can sit on it and the pollen grains can stick on the body of the insect, and therefore it can go to another flower and it can directly put the pollen grains on the stigma of that flower. Now if the flower is in a clove, or if the anthers and stigma are covered by the petals, pollination will not be possible there. So therefore, that is how you identify insect pollinated flowers. Or the flowers will have very bright colors, it will have good scent, it will have good nectar, and the petals will be very broad so that the insect can sit on it. All these are the nature of the flower to be pollinated by an insect. So that is how you need to identify, or by birds. So that is how you need to identify. So next is pollen germination on stigma through permanent slide or scanning electron microscope. So already a stigma which is being squashed and which is being stained will be kept. So you can see pollen grains on the stigma which has released or which has germinated and produced its pollen tube. So that you need to identify. Next is identification of stages of gamete development, and under that we have to identify the TS of the testis, that is the male testis, male reproductive structure, TS of the ovary, that is the female reproductive structure. So usually the slide, that is the testis or ovary of grasshopper or mice slides will be there in the laboratory, which will be kept, focused and kept. You need to identify what it is and write points on that. Next is meiosis is onion bud. That is true, that is grasshopper testis. Those cells will be already, permanent slides will be there, and different stages of cells will be in that. One will be focused by the examiner, or it will be asked by the examiner, that is metaphase. So we have meiosis 1 and meiosis 2. Under that, all the stages, leptotene, zygotene, pachytene, diplotene, diakinesis, all those stages, any one will be kept. So we need to identify what it is. Next, last one is we have TS of blastula, that is either permanent slide can be kept or a chart can be kept. So we need to identify it as blastula. So again, it is there for spotting. Again, we have Mendelian inheritance using seeds of different color. So Mendelian inheritance will be done very clearly in the theory class, and in the practicals it will be easy for you to segregate the characters. So that is using beads of different colors. Next is pedigree charts will be kept, wherein you have to identify the pedigree charts for different diseases and all that. You need to ask for certain genetic traits. It will be kept. Next is controlled pollination will be kept, wherein emasculation will be done. Emasculation is nothing but a flower will be kept wherein the anthers will be removed. So if the anthers are removed, it's called emasculated. That is the removal of the male reproductive part of the flower. That is an anther. So it's called emasculated. Then you can write it as emasculation. Otherwise, a plant will be kept and some labeling will be done to that. That is called as tagging. Otherwise, bagging. A plant will be kept with a flower and the flower will be covered by a bag. That is bagging. So you have to write about that, identify and write. Or they'll keep some disease-causing organism, say for example Ascaris. So Ascaris, usually chart or specimen will be kept. So Ascaris is nothing but roundworm. Then entamoeba slide will be kept. So entamoeba causes that is stomach-related disorders, entamoeba histolytica. Then the malarial parasite, that is plasmodium. This also slide or chart will be kept. And or any of the fungus, say for example the ringworm fungus will be kept. The permanent slides of that will be kept, or models can be kept. Anything can be kept. Charts can be kept. So you need to identify and write about it. Next, we have about symbolic association in root modules of leguminous plants. That is, say for example, rhizobium bacteria will be kept or will be focused. You have to identify. Or cuscuta plant. So cuscuta is a plant which is parasitic. So it is not green in color, it is yellow in color, it doesn't have any leaves, and therefore it doesn't photosynthesize because it's not green in color also, it doesn't have chlorophyll pigment. So how does it derive its food? It usually derives its food from other plants by growing on other plants, and sucking onto the nutrition of the other plants, eventually killing the host plant. So that is cuscuta. Even lichen also specimen will be kept. You need to identify it. Next is flashcards or models showing homologous and analogous organs. So homologous and analogous organs, some organs, they are similar in structure, but they have different functions. Some organs are different in structure, but they'll have the same function. So you need to identify the homologous and analogous organs. For this, charts will be kept for this. So this is about the practical. So out of those only questions will appear for the practical exam, and all those experiments will be conducted in the laboratory. Now the distribution of marks for your entire biology paper. As I told you, the entire paper is for maximum is 100 marks. So out of in 100, 70 is what you write in the main exam. That is, it is a theory paper. And 30 is what you do and perform and write in the practical exam, wherein even Viva will also be asked. Viva questions will also appear in the practical exam. Apart from that, you have to write the record, wherein record will carry 6 marks. You have to write the record and you have to answer to the questions asked. Totally, it will be for about 6 marks or 4 marks, and also for 4 marks. And also you'll have to submit a project, and you have to answer the questions or Viva questions related to the project. For that also marks will be allotted. All that will be done in the practical examination. So totally it is for your biology, totally you have to score 100 marks. Next, talking about the question paper, some general instructions will be given in the question paper which you need to follow. So all the questions are compulsory. Wherever there is internal choice, there only you can choose and write. Otherwise, all the questions are compulsory. And the question paper is divided into 5 sections. That is A, B, C, D and E. Part A, Part B, Part C, Part D and Part E. Now, all these are compulsory, and overall there is no choice, but somewhere in between, say for example, question number 22 can have a choice, question number 24 can have a choice. It's not that choice between question number 22 and 24. No, in question number 22 itself, 2 questions will be given. In that, you have to choose any one and write. So that is how internal choice will be provided. And wherever necessary, a neat label diagram should be drawn. Always remember, label the diagram, because however nice or beautiful you draw a diagram and you don't label it, then it is of no use. It's considered a 0. So therefore, draw a diagram and label it. Say for example, if it is for 5 marks, 10 labeling is compulsory. If it is for 3 marks, 6 to 8 labelings can be compulsory. So like that you have to draw and you have to label as well. Next, talking about the question paper design. So don't think that directly the questions will be asked like state board, how it is done. No, here the questions will test, it will test your analysis, it will test your capability of understanding the subject, how well you have understood the subject or the concept. All that, based on that it will be asked. So demonstrate knowledge and understanding, how you have understood the topic. Based on that, questions will be asked. That is indirect question, wherein you will have to think about it and then answer. So 50% of the question paper will carry questions pertaining to knowledge and understanding, and 30% will be application of knowledge or concepts. That is, again here, it can be direct questions can be asked or competency-based question can be asked. Next, 20% is analyze, evaluate and create. Here also paragraph kind question or graph-like questions and all will be given, and you have to see that, evaluate it and then answer. So that is for 20%. Very short answers will be asked. That is MCQs will be there and short answers will be there. 2-mark question, then long answers, 3 marks, then very long answers, 5 marks, like that questions will be asked. And internal choice are there. That is usually in part D and E and all that, internal choices will be there. Next, talking about the question paper pattern. As I told you, 5 sections will be there. That is section A, B, C, D and E. So section A is completely for 1 marks, wherein from question number 1 to question number 12, it will be multiple choice questions. And from question number 13 to 16, it is assertion and reason question, wherein topics will be given. That is, topic will be given and you have, or assertion and reason will be given, and out of the 4 options you have to choose. Next, section B is for 2 marks, wherein question number 17 to 21 is for 2 marks. In that, question number 21, there are 2 questions will be asked in question number 21. Say, for example, they'll write 21. So, they'll give one question, or they will write or here and give one more question. Meaning, out of these 2, any one you can write in question number 21. So, therefore, internal choice in question number 21 will be there. Next, section C is completely for 3 marks. That is question number 22 to 28. Here also, in question number 27, you will have internal choice. Next, section D, 4 marks. That is question number 29 and 30. So, these that is section D is completely case-based question. Here also in both 29 also you'll have an internal choice, 30 also you'll have an internal choice. Either they'll give a paragraph and ask you few questions, answer few questions out of it, or they'll give you a graph or something, they'll give a diagram and they'll ask you to answer questions pertaining to that. So, it is completely case-based question. Next, section E is 5 marks question, wherein question number 31 to 33, 3 questions also internal choice will be there. So, totally you will have 33 questions, wherein only in section B, C, so D and E you will have internal choices. That is only in one question. Only in question, that is section D, 2 questions internal choice, section E, 3 questions internal choices will be there. So, this is about the question paper pattern. And understanding of the topics is very, very important in CBSE, because blindly if you read, you cannot answer or you cannot score, because a lot of competency based and analysis kind of questions, understanding questions pertaining to your knowledge and skills will be asked here. So, that is why. So, concentrating each of the topic in the class is very, very important. So, this is about the question paper pattern. So, this was about the introductory class, wherein we learnt about the what are the textbooks, we got to know about the syllabus, the question paper pattern for the theory as well as the practical, and also we got to know about the course structure, be it the practical or the theory. So, this was about the session. So, I hope you found it useful. So, we shall meet again in the coming session, wherein we'll start with the concept classes discussing the topics under each of the chapters. So, see you in the coming session. Thank you.